Dear students, in the previous module we have already learnt about arson and how its investigation is done. You have already become familiar that the investigation of fire is done by expert from the relevant field that has expertise in fire investigation, and you are going to become one of these very soon. The investigating officer has the responsibility of securing the crime scene and rescue the injured person, which should be even more priority before doing the investigation. Proper documentation and climatic condition and direction of wind current is very significant in deciding about the evidence which you are going to collect from these scenes. Note making, recording, photography, sketching of crime scene are again the very important components and tasks of the investigating officer during such investigation, and you will be getting various important clues and evidences when you do this very carefully. Statements from eyewitnesses have to be recorded in written only. Searching of crime scene must be done by a proper way to avoid any kind of evidence destruction. Further on, the packing and preservation should be done according to their mandatory requirement, and you should be forwarding the collected exhibits to the forensic science laboratory for examination. And after the examination, the clue materials will be helping us in identification of the various accelerants or the sources which have been used to set the fire, as well as in the determination of the fire origin. We have significantly noted that V-shaped pattern and is very important, and you should make a clear note of that, take visuals and pictures, and that will be giving you the clue that where is the origin of fire which is there. And you have already noted that if there is a wind moving, then the V-shaped pattern gets distorted, which will be away from the direction of the wind. And you should be able to see that how this maximum damage is present at the origin of fire, which again will give you the clue about the origin of fire. Investigation officer must also inquire the facts about the motive or intention of fire, that is whether it is accidental or it is deliberate. We have already seen in the previous module that there can be so many motives which are associated with the intentional fire, and sometimes it is done by the juvenile persons who will be doing it just for fun, but then it again comes into a very serious outcome. Preparation of investigation or the opinion report has to be very clear and to the point and conclusive. And the final task of investigation officer is to deliver his or her expert opinion under the honorable court as witness. So dear students, we shall be continuing this study, and we shall study about the analysis of arson exhibits by instrumental methods. So let us begin the study of this module by knowing the learning outcomes. After studying this module, dear students, you shall be able to know how to carry out the extraction and distillation of fire samples, because once you have collected from there, how they will be further dealt up in the forensic science lab, that is what we will be studying here. Then how the chromatography works, and we will be specially focusing on the gas chromatography and the capillary gas chromatography. We shall also understand what are the ideal conditions for the instrumental examination that you have to follow for conducting these examinations or handling these instruments. So let us begin with the extraction of exhibits or the samples. Before the instrumental examination, the exhibit must be broken or cut down into small pieces, because you must have collected the big size of evidence also, arson crime scene. And furthermore, once you have cut down into small pieces, just take suitable amount of the sample for steam distillation. In general, approximately as a standard, 30 ml of distillate is required for instrumental analysis for the presence of flammable petroleum products such as petrol, kerosene, diesel, etc., which are commonly used as accelerants in the fire or arson cases, and we have already studied about these in detail, that is how the petrol, kerosene, diesel, what are the various parameters associated with them, the flash point and all, so that you will be able to identify whether you are dealing with the petrol or the kerosene or the diesel sample that you have collected. Now, focusing here, let us see how the extraction will be done. So, these petrol, kerosene, diesel, etc., which are the commonly used fire accelerants in arson cases, they are further extracted. And how that extraction is done? So, in order to conduct this extraction, you extract the distillate with approximately 30 ml of diethyl ether in about 2 to 3 extractions. Then combine the extracts and concentrate by evaporating the extract at room temperature to a small volume, that is, you have to reduce the volume to around 0.5 to 1 ml only. The standard samples of petrol, kerosene and diesel, by adding about 0.5 ml of each in a distillation flask, and blank samples are also prepared in a similar way for comparison. Alternatively, the direct extraction of the exhibits, standard and blank, can also be carried out with diethyl ether. So, let us try to understand how this extraction is carried out with this experiment. So, the concentrated extract which is so obtained is then used further for gas chromatographic analysis in both the cases, that is the one obtained from the crime scene and the other which are being used as the reference. Now, let us move further to the gas chromatography technique and how it is used in identification of these evidence that you have collected in context to their being petroleum products. Gas chromatography is used to detect the components based on selective affinity of components towards the adsorbent materials. The sample is introduced in the liquid or gas form with the help of a GC syringe into the injection port. It gets further vaporized at the injection port and then passes through the column with the help of continuously flowing carrier, which is known as the mobile phase, and it is mainly hydrogen which is commonly used, and this further gets separated or detected at the detection port with suitable temperature programming. This output, which is the chromatogram, is then visualized in the form of peaks in the computer, and further it can be attached to a printer to take the print of these chromatograms. Gas chromatography columns are of two designs, that is packed or capillary. The packed columns are typically a glass or stainless steel coil that is fitted with the stationary phase or a packing coated with the stationary phase. Capillary columns, on the other hand, are a thin fused silica or purified silica glass capillary that has the stationary phase coated on the inner surface. So, out of these two, let us first study about the gas chromatography. The gas chromatography is a powerful and widely used tool for separation, identification and quantization of components in a mixture, and this is commonly abbreviated as GC. In this technique, a sample is converted into its vapor state and a flowing stream of carrier gas, often helium or nitrogen, sweeps the sample into a thermally controlled column. A gas chromatographic system consists of a regulated and purified carrier gas source which moves through the sample through the GC, an inlet which also acts as a vaporizer for liquid sample, a column in which the time separation occurs, a detector which responds to the components as they occur or are detected by the changing of its electrical output, and then data interpretation of some sort. So, as you can see in this schematic diagram for the gas chromatography, all the important components that I told you just now, we can see them in this diagram. This cylinder is represented that this is containing the carrier gas, and then through this there is a flow controller to manage the volume of the gas flowing, and then there is a sample injector from which the sample will be introduced, and this is the same sample that you have prepared via the extraction using ether, or you will use the reference sample one by one. And once you have injected in this, you can see this is the circular column, and this is there inside the oven. And if you look at the significance of oven, you can control the heating condition and it will vaporize the liquid sample if you have added. And because it is a gas chromatography, the sample will be there now in the form of the gaseous form. And once it goes from this outside to the detector, moving via the adsorbent, then in the detector it is detected and it is then displayed in the form of the peaks either in the computer screen, and which can be further connected to the printer, and you can take the printouts of these chromatograms. Now, if you look at the experimental conditions which are used as reference for conducting these experiments, suitable volume of the concentrated extracts of the exhibit, standards and blank samples are injected into a gas chromatograph with these conditions. Let them understand one by one. First of all, if we talk about the column, PAC SC30 is used, which is Epigeon L or its equivalent column, and this can be used for the separation of petroleum products and similar other compounds. If you look for the detector uses flame ionization detector, which is abbreviated as FID. The carrier gas used is either nitrogen or helium. The flow rate which is kept is 30 milliliters per minute, and the over temperature is programmed, and this again is variant for different materials that you are using. So, if it is for petrol or gasoline, it starts at 70 degrees centigrade, it is kept isothermal for 5 minutes and increases with 50 per minute up to 120 degrees centigrade. So, the range is you keep it from 70 to 120 degree centigrade. Now, if the same process is being done for kerosene, then the temperature starts from 90 degrees centigrade and continues at 10 degrees centigrade per minute up to 220 degrees centigrade. The range is 90 to 220 degrees centigrade and hold is for 5.5 minutes at 220 degrees centigrade. The same if it is done for diesel, it starts from 90 degrees centigrade and continues at 10 degrees centigrade per minute up to 240 degrees centigrade, that is 90 to 240 degrees centigrade is the range, and hold is done for 10.5 minutes at 240 degree centigrade. After the oven temperature programming, let us now understand the detector temperature setting. At the detector level, the temperature are set. For petrol it is 170 degrees centigrade, for kerosene it is 230 degrees centigrade, and for diesel it is 240 degrees centigrade. Moving to next, the injector temperature. The injector temperature are set this way. For petrol it is 170 degrees centigrade, for kerosene it is 230 degrees centigrade, and for diesel it is 240 degrees centigrade. And dear student, why we are looking for different temperature setting? It is because you have already studied in the petroleum fractionation, when you have separated the crude petroleum, the different fractions are obtained based on their different boiling points. So, for converting them into the gaseous form, you need to have the different temperature which are around their boiling points, and that is how you have putting it in these variant ways depending upon their composition that you are already aware. For example, let us take the case of petroleum product for which the operating conditions are as such. The column is 6 by 0.125 SS. The packing is OV1 SE30 or Epigeon L or its equivalent column, which can be used for the separation of petroleum products. Detector is the FID, that is flame ionization detector. Carrier gas is either nitrogen or helium. The flow rate is kept as 30 milliliters per minute. Oven temperature is programmed for 60 degree to 250 degrees at 4 degrees centigrade per minute variation. Detector temperature is 275 degree centigrade and injector temperature is kept at 250 degree centigrade. So, in the gas capillary chromatography or capillary gas chromatography, there are capillary columns which are thin fused silica, that is purified silicate glass as capillary, which is used as a stationary phase coated on the inner surface. Capillary columns provide much higher separation efficiency than the packed columns, but are more easily overloaded by too much sample. For petroleum product analysis, we have to again maintain some specific condition as we have done for the gas chromatography, and that we can see here. The operating conditions are: the column used is 50 to 100 m glass capillary into 0.01 ID. Packing is OV101 or its equivalent column. Detector which is used is the same, which is flame ionization detector, FID. The carrier gas here is nitrogen. The flow rate is kept as 1 milliliters per minute. The oven temperature is programmed for 60 degrees centigrade to 250 degrees centigrade at 2 degrees centigrade per minute variation. Detector temperature is kept as 275 degrees centigrade and the injector temperature is kept at 250 degree centigrade. So, if we talk about the schematic diagram of capillary gas chromatography, it is almost the same as gas chromatography. That is, there also we had the carrier gas, here also we will have the carrier gas from which the flow controller can manage its input, and then you will be injecting the sample from the sample injector, and there will be column which is inside the oven, but here the nature of the column will be differentiating. That is, you will have the adsorbent which will be adsorbed inside these columns and not packed, unlike the gas chromatography. And then once this is detected at the detector, there will be display of the peaks as the chromatogram in the computer, or you can take the printout. So, after understanding these two instrumentation method and how the samples have to be prepared for them using the extraction with the help of ether, let us now look how the chromatogram of petrol and diesel will look like. Because once you are aware with the chromatogram of how the petrol or diesel look like, which will be the reference sample, what chromatogram you have obtained from the sample, you can simply compare and tell whether it is of the petrol or it is of the diesel, and so in a way you will be able to identify, and you can also do the quantitative study in knowing the quantity of the sample. So, shown here is the typical gas chromatogram of diesel. You can see here it is having so many peaks, and if you notice why there are so many peaks, because diesel is not a single organic compound, it is a mixture of so many organic compounds, and corresponding to each of them you are having the peak. So, this is how the gas chromatogram of diesel look like. On the other hand, if we look at a typical gas chromatogram of kerosene, then in kerosene you can see the peaks are of different shape, but here also there are so many multiple peaks which are there, and another thing is the values of the peak, that where they are appearing, that has to be matched with the sample that you have collected from the crime scene, so that you can say whether it is kerosene or not. And that too different grades of kerosene can be compared, that which particular kerosene has been utilized. Next is the typical gas chromatogram of gasoline, and here again you can see there are so many peaks, but here the peaks are more intense and sharp and not merged, like they were giving you a bigger peak and in between they were smaller peaks arising in the case of diesel. So you can clearly see there is so much difference which is there in the peaks of these three. That is, let us see these gas chromatograms for the three again. This is how the gas chromatogram of diesel looks like. This is how the gas chromatogram of kerosene looks like, and this is how the gas chromatogram of gasoline looks like. So you can clearly see the difference between the peaks, their values, their location and the nature which is there, and that all has to be compared when you are taking a sample and finding out its gas chromatography and taking out its chromatogram. The similar treatment can be done for the capillary gas chromatographic technique, that is, you will be having these samples which you will be running through, which you know it is either petrol or kerosene or diesel, and then you have your sample which you have collected from the crime scene. You have to simply compare that sample with these reference samples, and that comparison will give you clear idea whether the sample you have collected from the crime scene is of petrol or diesel or kerosene. So dear students, let us summarize what we have learnt in this module. We have learnt how the extraction or distillation of exhibits or sample is done with the help of diethyl ether, and recall you have to do it 2 to 3 times, and then you have to collect these samples, concentrate them, and that is how you will be able to use this sample further for the chromatographic techniques. And the chromatographic techniques we have explained in this module are the GC and the capillary GC. Chromatography is a separation technique which is used to separate the complex mixture in all forms, that is solid, liquids and gases. This you should keep in mind while you are conducting these experimentation. And if we talk about the column is of two designs, packed or capillary in terms of gas chromatography, and when you use it as packed, then it is the gas chromatographic technique, and when it is in the form of capillary, where the coating of the adsorbent is inside that column, then it is the capillary GC term used. And the separation is recorded in the form of chromatogram, and analyzing these chromatogram with the reference chromatograms, you can clearly get the idea what are the various components which are present. The essential component of a gas chromatography and capillary gas chromatography we have studied are the injection valve, from where you will be injecting the sample. There is a mobile phase which is gas in both of these, that is the gas chromatography as well as the capillary gas chromatography technique. Then there is a stationary phase which is there as column, and the column is differing in case of gas chromatography and the capillary gas chromatography. Then there is a oven which is there inside, you have the circular columns, and this oven helps to maintain the temperature and helps the injected sample to be vaporized into the gaseous form. And then there is a detector from which you can detect these peaks for the various different components as and when they reach the detector, and this output, it can be seen on the computer screen as the various peaks, and it can also be printed out. And we have also seen how the various chromatograms typically look like, but it is always advisable that whenever you are conducting any such study or the instrumentation, then always you should carry out one with your sample that you have collected from the crime scene as evidence, and then you should compare it with the reference sample, always fresh, because if you have set the conditions differently, then these peaks may get shifted to lower or higher values, and that may confuse you. So it is always advisable that you compare your sample that you have taken with the reference ones, all the chromatograms freshly printed out through the GC or the capillary GC instrumentation techniques. And if we talk about GC and capillary gas chromatography, they are the most ideal tools for the petroleum product analysis. So after being aware about how these samples you have collected from the arson scene in the forensic science laboratory with the help of GC and the capillary GC, in the next module we shall understand how to use all this knowledge for management of arson cases. We shall study about the various occupational standards for crime scene management, which include the standards and the assessment protocols and how the management of these cases is done. We shall also study how to deal with arson crime scene management very effectively. We shall comprehend the various occupational standards and protocols used during arson crime scene investigation or management, and as a forensic scientist, what shall be your duties during the investigation management of these arson cases.